It's important to clearly outline your inclusion-exclusion criteria. This is always a balance and probably one of the most important parts of the protocol. This can either kill your trial or you can be a hero and actually recruit ahead of time, which is a rare event in clinical trials. So think carefully about who's your study population and what inference you'll make at the end. If you make it so narrow, you're going to hurt your enrollment, and when you finish, if ever, the study, it's going to be difficult for others to then generalize that across perhaps the broader disease population. On the other hand, if you make your inclusion-exclusion criteria so broad, you may have so much variability in your patient population that you may not be able to get an answer to the question, or you may not be able to see the signal if one exists. So it's always a balance between those two objectives. You should carefully describe your intervention, where it comes from, how it's formulated, how it's going to be administered. Think about what other medications or therapies a person may be allowed when they're in your trial and describe that. Is there anything that's prohibited? Is there anything that they can take but limited to a certain class? Or because you have to think about what potential interactions they may have with the intervention of your study. A clear detailing of every study procedure that's going to be done and the schedule on which it's done, how you're going to report adverse events, and then how you're going to be monitoring the drug or the biologic or the intervention in terms of, is it being administered as it's supposed to be? Are there enough supplies in the sites? Are you monitoring the patients? And is the site being monitored in terms of their ability to keep up with the study requirements? In the statistical considerations, the primary and secondary objectives should be clearly stated with the sample size calculation, and that so that you know you have adequate subjects to meet your objectives. In a large phase 3 trial, there's often interim monitoring both for safety and efficacy, as I spoke earlier about, in terms of should I stop the study early? Is there enough evidence to convince the broader audience that what I have here is likely real and not just due to chance? And then the statistical analysis plan. In the appendice, often you might have a very detailed exam flow chart that would list each visit and every procedure that's going to be done at that visit. And then template-informed consent. So each site has to do their own consent per their own institutional review board, but the main protocol should provide the template consent so there's some uniformity in the way the subject is being described to the subjects. One of the things that I've seen in some protocols are people like to put in rosters, sometimes those really detailed study-specific procedures. And those are better left to other documents because they're subject to change. So things that you think are going to change a lot, that are very detailed, you don't want to put it in your protocol because every time you change that, you need to amend your protocol. And that starts you down the regulatory avenue, which takes a lot of time and is not always going to be conducive to efficient trial conduct. Most protocols are going to require amendments, and they should. You never want to change anything in your protocol and not document it.